हेलो डियर लर्नर्स, वेलकम टू माय चैनल एम्ब्रेस बायोटा। आज के लेक्चर में हम ब्रायोफाइट्स के इम्पोर्टेंट पॉइंट्स को डिस्कस करेंगे। बिफोर दिस हमने फंजाई का वन शॉट करा है। अगर आपने वो वीडियो नहीं देखी है तो आप उसे देख सकते हैं। दिस इज द पार्ट 5 ऑफ सीरीज जिसमें हम HPU M.Sc बॉटनी एंट्रेंस के लिए प्रिपरेशन कर रहे हैं। आज के लेक्चर में हम ब्रायोफाइट्स से रिलेटेड सभी इम्पोर्टेंट पॉइंट्स को कवर करेंगे। The study of bryophytes is known as bryology. Term bryophytes is proposed by Robert Brown. Father of bryology है Hedwig. And the father of Indian bryology is S.R. Kashyap. Largest bryophyte is Dawsonia and the smallest bryophyte is Zopsis. Bryophytes are the first land plants. But these are not completely terrestrial. Bryophytes को पानी की requirement होती है for their fertilization. That's why the bryophytes are called as amphibians of plant kingdom. Bryophytes are the 8 tracheophytes. It means they lack the vascular bundles, so called as avascular cryptogams. It means that vascular bundle lacking non-flowering plants. These are the first embryophyta and the first archegoniate. Archegoniate is referred to the female sex organ which is known as archegonia. And the important point is that the Western Himalayas is known as gold mines of liverworts. Next is the habitat of bryophytes. These are mostly terrestrial. The example of aquatic bryophytes are Rixia species and Sphagnum. Epiphyllous species are Radula. Example of epiphytic bryophytes are Dendroceros and Frularia. Porella is lithophilic bryophyte. And Polytrichum and Tortula lives in xerophytic conditions. Talking about the nutritional needs, bryophytes are mostly autotrophic. Saprophytic species are Boxbomia and Cryptothalus. In the thallus of bryophytes, the main plant body is gametophytic and the body is thalloid, that is not differentiated into true root, stem and leaves. Bryophytes are heteromorphic, that is alternation of generation seen as there is two phase. This shows the haplodiploidic life cycle, in which the haploid phase is dominant over the diploid phase, that is gametophytic phase is dominant over the sporophytic phase. The gametophytic phase where the sex organs and produce the gametes. It is independent, green in color and is long-lived. Whereas the sporophytic phase is the diploid and produce spores by meiosis. The sporophytic phase is completely dependent on the gametophyte and we can say that it is parasite over gametophyte. This point is important. Talking about the gametophytic plant body, it can be prostrate or erect and aerial. Prostrate and dorsiventral plant body is seen in hornworts and liverworts, whereas gametophyte is erect and aerial in mosses. Mosses में further देखें तो gametophyte is of two types. First is protonema and second is leafy. The prostrate gametophyte is known as protonema and the erect is leafy. Many times the question is asked that in which class of bryophytes protonema stage is seen. If we closely see the plant body of the bryophytes, we can say that the dorsal and ventral surface are morphologically different. The dorsal surface is smooth whereas the ventral surface is rough. Ventral surface is rough due to the presence of rhizoids and scales. Rhizoids are root-like structure and help in absorption of minerals and water. And the scale helps to prevent the water loss. The distribution of rhizoids is different in different classes. In Rixia and Markensia, it is on either side of the midrib. And in Anthoceros, there is no proper distribution of rhizoid. It is of scattered type. Rhizoids may be unicellular and unbranched or multicellular branched with the septa. Unicellular and unbranched are found in liverworts and hornworts. Multicellular branched rhizoid with oblique septa is seen in mosses. Rhizoids are further divided into smooth-walled rhizoids and tuberculated rhizoid. As the name suggests, smooth-walled rhizoid में किसी भी तरह का कोई depression present नहीं रहेगा. But the tuberculated rhizoid is folded into the peg-like manner and form the appendages. Rixia and Markensia possess both smooth-walled as well as tuberculated rhizoid. But in Pallila and Anthoceros, only smooth ball rhizoid are present. राइजॉइड के बाद हम बात करेंगे स्केल्स की। स्केल्स जो हैं वो थैलस के वेंट्रल साइड में प्रेजेंट हैं एंड दीज आर मल्टी सेलुलर इन नेचर। Scale is a feature which is restricted only to the order Marcantiales. Marcantiales के हमारे important members हैं रिक्सिया एंड मार्केंशिया। डायग्राम में देख सकते हैं कि रिक्सिया में स्केल जो हैं वो ओनली सिंगल रो में प्रेजेंट हैं, बट इन मार्केंशिया स्केल्स आर प्रेजेंट इन 2 to 4 rows on the either side of the midrib. Both the ligulate as well as appendicular scales are present here. I also added a diagram for your better understanding. The cell wall of bryophyte is cellulose rich. Reserve food material in the bryophytes is starch and pigment are chlorophyll A, B and carotenoids just like other plants. थैलस की बात करें तो liverworts में थैलस को डिफरेंशिएट करा है into upper photosynthetic zone and the lower storage zone. इसके अपोजिट hornworts में हमारे पास थैलस undifferentiated है. Here the photosynthetic cells are all over the thallus body. Talking about the liverwort thallus, the Rixia and Markensia has different thallus structure. In the thallus of Rixia, the photosynthetic filament is unbranched. But in the thallus of Markensia, this filament is branched. This is a important distinguishable point between these two. In between photosynthetic filament, the air spaces are present. Beneath photosynthetic zone, there is the storage zone. राइजॉइड की बात करें तो वो हमने पहले ही देखा कि रिक्सिया और मार्केंशिया में स्मूथ एंड ट्यूबरक्युलेटेड दोनों राइजॉइड प्रेजेंट हैं, बट द डिफरेंस लाइज इन द स्केल. इन रिक्सिया द स्केल्स आर लिग्यूलेटेड एंड इन मार्केंशिया द स्केल्स आर लिग्यूलेटेड एज वेल एज अपेंडिकुलर. यहाँ से हमें रिक्शिया और मार्केंशिया के दो डिफरेंस क्लियर होते हैं. फर्स्ट वन इज द प्रेजेंस ऑफ अनब्रांच्ड photosynthetic filament in Rixia, whereas it is branched in Markensia. The second is the ligulated scale of Rixia and appendicular scales of the Markensia. हॉर्नवर्ट के थैलस की बात करें, इट्स थैलस इज अनडिफरेंशिएटेड, दैट इज नो फोटोसिंथेटिक और स्टोरेज जोन इज देयर. इंपॉर्टेंट फीचर इज द प्रेजेंस ऑफ नॉस्टॉक कॉलोनी इन द थैलस ऑफ एंथोसिरोस. अब हम देखेंगे ब्रायोफाइट्स के सेक्स ऑर्गन्स के बारे में. सेक्स ऑर्गन इन ब्रायोफाइट्स आर मल्टीसेलुलर एंड जैकेटेड. मेल सेक्स ऑर्गन की बात करें उसे एंथ्रीडियम कहते हैं, वेयर एज फीमेल सेक्स ऑर्गन इज नोन एज आर्किगोनियम. Antheridium is single in liverworts and in group in mosses and hornworts. The antheridium is stalked, globose-like and has sterile jacket. Fertile cells in the antheridium is called as androcytes. These androcytes produce the male gamete antherozoids. The antherozoids are biflagellated structure. Female sex organ is known as archegonia. It is flask-shaped. It has 3 parts: neck, venter and egg. The important thing is that the location of fertilization is inside venter. फर्टिलाइजेशन देखें तो फर्टिलाइजेशन के लिए वाटर की रिक्वायरमेंट रहती है. इस तरह की फर्टिलाइजेशन को हम ज़ोइडोगैमस फर्टिलाइजेशन कहते हैं. एंड द फर्टिलाइजेशन अकर इनसाइड द venter, so zygote is produced inside it. Embryo also develop inside the venter, which divide to form the sporophyte. The sporophyte is a diploid structure and it is short-lived. Sporophyte is parasite over the gametophyte plant. It can be total parasite or partial parasite. Sporophyte is total parasite in case of liverworts and the partial parasite in hornworts and mosses. If we closely observe the structure of sporophyte, we can see the 3 parts in the sporophyte. These are foot, setae and capsule. लेकिन अगर हम रिक्सिया की बात करें तो रिक्सिया के sporophyte में only capsule is present. It means the sporophyte of Rixia lacks foot and setae and is the simplest sporophyte. This point is important. In Markensia, Porella and Pallila, capsule, setae and foot, all 3 structures of sporophyte are present. Anthoceros के sporophyte में seta absent रहता है. One of the most important point is that instead of seta, here the intercalary meristem is present. As we all know, meristems are the growing cells, so these grow non-stop and give a very bulky appearance. That's why it is known as hornworts. Most complex sporophyte in bryophytes is seen in case of mosses. Capsule contains the spore mother cells. These undergo meiosis and produce the spores. In bryophytes, only one kind of spores are produced, so bryophytes are homosporous. About spores, the most interesting point is spore dispersal. It occurs by different mechanisms in different classes. In liverworts, there are three mechanisms of spore dispersal. First one is nerve cell found in Rixia. These are called as forerunner of illators. These nerve cells are the sterile cells of the capsule and provide food for the developing spores. They also take part in the spore dispersal. Illators are seen in Markensia and Porella. Illators are also enclosed within capsule, and on maturity the protoplasm of illators become dead and hygroscopic. Due to this they absorb water and when burst help in the spore dispersal. Illatophores are seen in Pellilla and Riccardia. Illatophores are collection of illators. In hornworts, the pseudo-illators are meant for spore dispersal. Whereas in mosses, the presence of peristomial teeth is there. These are usually 32 in number. But the special mechanism of dispersal is seen in Sphagnum, where the spore dispersed by air gun mechanism, and in Polytrichum, where the spore are dispersed by sensor mechanism. This is the diagram of life cycle of bryophyte. Bryophytes are haplodiploidic in their life cycle. You can see here the gametophytic as well as sporophytic phase clearly. The gametophytic phase is large and is dominant over the sporophyte phase. Next is the reproduction in bryophytes. Bryophytes reproduce by oogamous type of sexual reproduction. Bryophytes में सेक्स ऑर्गन डोर्सल सरफेस ऑफ थैलस में रहते हैं इन लिवरवर्ट्स एंड हॉर्नवर्ट्स. मोसिस के केस में ये सेक्सुअल ऑर्गन्स लीफी स्टेज ऑफ गैमीटोफाइट में पाए जाते हैं. मेल सेक्स ऑर्गन हमारे पास एंथेरिडियम है. एंथेरिडियम कैन बी सिंगल और इन ग्रुप्स. It is single in liverworts and present in group in hornworts and mosses. Iridium की origin की बात करें तो it can be endogenous and exogenous. Endogenous origin, the male sex organ developed from the hypodermal cell. It is seen only in the case of hornbirds. This point is to be noted. In exogenous, the male sex organ arises from the superficial cells. It is present in liverworts and mosses. थ्रीडियम की लोकेशन देखें तो इट कैन बी एम्बेडेड इन थैलस और मे बी प्रोजेक्टेड आउटवर्ड. इट इज एम्बेडेड इन केस ऑफ रिक्सिया एंड एंथोसिरोज. एंड थ्रीडियम इज प्रोजेक्टेड आउटवर्ड इन केस ऑफ मार्केंशिया एंड फनेरिया. In Markensia, it is present in male receptacle known as enthridiophore, and in Phaneria, it is surrounded by perigonial leaves. If we see the enthridia, it is surrounded by a sterile jacket. Inside the jacket, the fertile cells are present called as androcyte mother cells. These mother cells undergo the diagonal division and result in the formation of two triangular androcytes. Androcytes result in the formation of antherozoids, which is male gamete. The male gamete is comma-shaped biflagellated structure. Two flagella are anterior in position and is of whiplash type. The female sex organ is multicellular and jacketed, and it is first archegoniate, where the female sex organ is known as archegonia. एंथ्रीडिया का ओरिजिन एंडोजीनस और एक्सोजीनस दोनों टाइप से था, लेकिन फीमेल सेक्स ऑर्गन का ओरिजिन इज प्योरली एक्सोजीनस. स्मॉलेस्ट archegonia is seen in case of Rixia and largest in case of Funaria. Talking about the location, it can be embedded in thallus or is projected. In Rixia, it is partially embedded, and in Anthoceros, it is completely embedded in thallus. The main feature of the archegonia of Anthoceros is that it is non-jacketed and is protected by a cell of thallus itself. The projected archegonia is seen in case of Markensia and Funaria, just like male sex organ. In Markensia, it is found in female receptacle known as archegoniophore. In Funaria, is surrounded by parietal leaf. If we talk about the structure of archegonia, the upper cylinder part is known as neck and the lower swollen part is known as venter. Neck is surrounded by only single layer of sterile jacket. It is covered with four-celled lip or operculum. Sterile jacket of the neck, there can be 6 vertical rows of neck cells or 5 vertical rows of neck cells. The 6 vertical rows is common and the 5 vertical rows is seen in Pellilla and Porella. इसका basal solen part जिसे venter कहा जाता है is also surrounded by sterile jacket. But the difference is this jacket can be venter in single layer, 2 layered or 2 to 3 layers. Single layered is most common seen in Rixia, Markensia and Anthoceros. Two-layered sterile jacket is seen in the Benter of Neria and up to 3 layer is seen in order Jungermanniales. For better understanding of this concept, I also added a diagram here. The cavity in the venter contains the upper smaller venter canal cell and the lower larger female gamete known as egg. And the cavity of the neck contains the neck canal cells. Only 4 neck canal cells is found in Rixia, that is why Rixia has smallest archegonium. 6 to 8 neck canal cells is seen in most of the cases. The most important is 18 neck canal cell is present in Funaria, that is why it has largest archegonia among bryophytes. The fertilization is zooidogamous, that is occurs in the presence of water. The female gametes secrete chemical like malic acid. Male gametes swim in the water and enter into the venter. Union of male and female gamete occur in the venter and the zygote is produced. Act of fertilization stimulates the venter to form a covering around the zygote and this covering is known as calyptra. This is a very important question about calyptra as it is a gametophytic tissue which is formed by the venter. Most of the time the polydy of the calyptra is asked or it is asked that which type of gametophytic tissue is produced by venter. Zygote further divide to produce the sporophyte which consists of foot, setae and capsule. The foot is embedded in gametophyte and derive nutrition from them. Seta help in the conduction of nutrition from the foot to capsule. Capsule cell further divided and form the outer and inner layer. This concept is very much important that the outer layer of the capsule is known as epithecium and the inner layer is known as endothecium. Epithecium divide and form the jacket of capsule. The important point is that the endothecium is converted into archisporial cells. The jacket of capsule formed by the epithecium can be 2-layered, single-layered or multi-layered. It is multi-layered in case of Anthoceros and Funaria. This point is important. Endothecium which is converted into archisporial cell. What exception lies in case of Anthoceros? After converting into the archisporial cells, it continues divide and produce the sporogenous tissue which contains the spore mother cells. The spore mother cell undergo meiosis and produce the spores. All the sporogenous tissue produce the spores in case of Rixia. But in Rixia crystallina, some sporogenous tissue becomes sterile and form the nurse cells. Nurse cell provide nourishment and helps in spore dispersal. Markensia, the sporogenous tissue is half fertile and half sterile. A fertile portion forms the spore and sterile portion forms the illators. Illators also help in spore dispersal. Talking about the sporogenous tissue of Anthoceros which is not formed from the endothecium. In Anthoceros, the entire endothecium becomes sterile and converted into columella. The sporogenous tissue is formed from the epithecium. This exception of sporogenous tissue is of much importance. In Anthoceros, the 1/3 part of the sporogenous tissue is fertile which forms the spore. And the 3/4 part is sterile which forms the pseudo-illators. Pseudo-illators are like illators but here the spiral thickening is absent. In the Funaria, the most part of endothecium is converted into columella and sporogenous tissue is formed from the outer layer of endothecium. When we move from the lower to higher bryophytes, that is from Rixia to Funaria, the sporogenous tissue start becoming sterile. Most important point of the sporogenous tissue is that the Anthoceros is the only bryophyte in which the sporogenous tissue is not formed from the endothecium. Instead of endothecium, it is formed from the epithecium. This is all about the important points related to bryophytes. The next lecture we will discuss about all the important genera of bryophytes. If you find this video helpful, subscribe the channel. My name is Aina Sharma and thanks for watching.